Okay, so this is genetics chapter 21, and we're going to take a look at some principles of inheritance. And so when we um, look at this, the first thing we get into in genetic inheritance is the difference between genotype and phenotype. So genotype basically is essentially all the genes of an individual. So essentially genes are segments of DNA that code for a particular trait, oftentimes, not always, but oftentimes a protein. And so there's a specific location on each chromosome where the genes are located. That's called a locus, loci for plural. So this is a location of the gene on a particular chromosome. Now each gene has different versions of it. So alternative forms of a particular gene is called an allele. For instance, if you take a look at the trait eye color, you might have an allele for blue eyes and one for brown eyes. So two different versions of the same gene. Oftentimes, when we take a look at alleles, they can be classified according to their strength. For instance, one allele can be thought of as dominant and the other one as recessive. And a dominant is typified by the fact that they can mask the expression of the recessives. So the recessives kind of hide when they're in combination with the dominants. And as a result, if you only have one copy of the dominant, then you'll be able to see that expressing, that dominant allele that is, expressing through to the phenotype. However, a recessive, because it's hidden by the dominant, you're only going to be able to see it if you have both alleles of the recessive. So for instance, a person with blue eyes, which is a recessive trait, has to have two recessive traits for blue eye in order to see the blue eye. Otherwise, if it's a combination of the two, then it's going to look like a brown eye. And so by convention, we'll oftentimes denote or notate the dominant allele as an uppercase letter and recessives as lowercase. For instance, for albinism, normal pigmentation is capital A, the no pigmentation is little a. And so when we are getting these traits from our parents, we receive one allele, one chromosome from each parent. And so we'll have one allele from one parent, one allele from the other parent. Now, there's different types of genotypes that you can create from these pairings. So if you have two dominant alleles, it's what we refer to as homozygous dominant. So homo means one. Zygous is referring to the zygote, which is your single-celled stage. And so in this case, you'd have two uppercase, big A, big A. Basically, you're getting a dominant trait from the mom, that is the egg, and the dominant trait from the dad, which is the sperm. You can also get homozygous recessive, in which case both alleles are recessive. And then there's heterozygous, where you have a combination of each, where you have one recessive and one dominant. That would be considered a heterozygous. Hetero means different, zygous, different eggs. And so when you take a look at our traits, you can see here, it's basically a mixture of how mom and dad basically advance forward the alleles that they produce and that they give to us during development. So for instance, if dad advances the dominant A sperm with mom's dominant A egg, then we will be homozygous dominant. We'll have basically dark hair. Now, if we have two recessives, so that is to say, if dad gives a sperm with a little a and mom with a little a, that means we'll be homozygous recessive. And so we'll have the recessive trait, so that is light colored hair. And then, of course, if mom gives one dominant and dad gives a recessive or vice versa, then that means we're going to be a heterozygote, which basically means, for the most part, because of the dominance of the dominant allele covering the recessive, we oftentimes will look very similar to the homozygous dominant. And so that basically then would be our phenotype. So our phenotype is essentially the physical manifestation of all of these traits. And oftentimes the genotype adds directly to the phenotype. So that's to say that what your genotype is going to be essentially predicting what your phenotype is. So your phenotype is essentially the lump sum of all of the dominant and recessive and all those sorts of traits that are basically in your entire genome. That is all the genes that you have in your body. Now, we can take a look at genetic inheritance by either looking at either inheriting one trait or two traits. So if we're inheriting one trait and we do a one trait cross, then it's what we refer to as a monohybrid. So we're hybridizing one trait. And so this will be taking a look at a single set of alleles for a single characteristic. We can also do a two trait cross, which basically takes a look at two different characteristics at the same time. And so we'll have like maybe hair color and eye color at the same time or something of that nature. And so this will be two different characteristics. Both of these crosses are necessary for the gametes to be able to move forward to the next generation. So basically, you'll get your determination from the gametes that your parents give you. So you are what your parents have given you. So when the gametes fuse, each individual, that is us, we start off with 46 chromosomes. That's what's called our diploid number. And so we have 46 chromosomes. We have two pairs of each 23 pairs. So we have 23 pairs. So we have two of each. And those are important because basically when we take a look at the 46, because we have two pairs, we get one set from mom and one set from dad. That's why we have 46. And so those individuals are what's referred to as homologs, right? So for instance, the first chromosome, the mom one and the dad one are essentially homologs of each other. And so during meiosis, these homologous chromosomes will separate, and that gives us basically a cell of 23 chromosomes. Now, when you take a look at one trait crosses, one of the things we can try to do is predict the chances of having a particular child with a certain genotype, right? And so if we take a look at this monohybrid cross, when we take a look at freckles, for instance, we'll take a look at what the likelihood is of having a child who has freckles. So for instance, if we take two parents that don't have freckles, first of all, you have the dominant allele for freckles, and of course you don't know what the other one is. It could be also dominant, it could be recessive, you don't know. And then you have the recessive allele for no freckles, right? So big F and little f. And so if both of the parents have no freckles, and freckles is a dominant sort of thing, then that means both parents have to be recessive because they don't have freckles, because we just defined that system here, right? Because we said that basically the dominant allele is for freckles. So if you have freckles, it's a dominant trait. So if both parents basically don't have any freckles and they're both recessive, that basically means that each parent can only give forward a recessive allele, in this case, the no freckle allele. That's true for both parents. And then they can only basically donate that to the offspring, which will also have no freckles. Okay, so if you're taking a look at freckles here, you can see this individual who has freckles is going to be either homozygous dominant for freckles or heterozygous. Either way, you're going to see freckles. This individual who does not have freckles basically will be homozygous recessive. Now, notice the frequency of the phenotype in the human population does not necessarily match what's actually the dominant or recessive, right? So one could argue that probably freckle occurrence in the human population does not occur with as much frequency as you would expect. So just because it's dominant doesn't mean that everybody has it, right? It all gets kind of different, how it sort of segregates throughout the population. So if you take a look then at another cross, so in this case, you're going to take a man who has freckles and he's homozygous dominant, so we know that one. And he is going to have a child with a woman who has no freckles, so that one is going to be recessive. Then when these two pair off, the dad can only give the dominant freckle allele, and the mom can only give the recessive no freckle allele. And so when you combine these two together, it creates a heterozygote child who would be dominant for freckles and would have freckles. So if one of your parents have freckles, or if you have freckles, then take a close look at your parents. One of them has freckles as well. And so in order to kind of keep track of this, we have a tool, a little graphical kind of a tool, organizational tool that we use called the Punnett square. That helps us to be able to predict the outcomes of crosses when we're taking a look at these sorts of problems where we're trying to figure out what's the likelihood of getting a child with a particular genotype. So what we do basically is we take a look at the possible alleles for the sperm. So how many different types of sperm can we generate? Same thing for the eggs. How many different types of eggs can we generate? And then we take a look at all the different possible combinations of each of these gametes coming together. For instance, if we take a cross of two heterozygotes, so in this case, we're sticking with freckles, right? So we have two heterozygote parents. Then you have each parent has two different gametes they can give, right? So for instance, they can either give the big F or they can give the little f. It depends. Now, if you take a look then at Punnett square and use kind of Punnett square, then what we can say is like, okay, each parent has the ability to eventually give either a big F or a little f. Same thing with this one, can give a big F or a little f, not both, just one. And so what happens if that occurs? So, well, first of all, this particular egg right here, if it pairs with this sperm, will give you a homozygous dominant. If this egg pairs with this sperm, then it'll give you a heterozygote. If this egg pairs with this sperm, it'll give you another heterozygote. And if the little f pairs with the little f, it'll give you a homozygous recessive. So then, knowing then that the big F is dominant and 4 freckles, then basically what that means is that you will expect to have 3 freckled individuals relative to 1 no freckled individual. Now notice, these are the possible outcomes. It doesn't necessarily mean that you're going to have them. It just means these are just the possible outcomes. Okay. So if you had, say, for instance, this is the possible outcomes for each child. So if this couple had like 4 children, it doesn't mean they're going to have 3 freckled and 1 no freckle. It just means that each child has a 3-to-1 chance of being freckled. So then if you take a look at the genotypic versus the phenotypic ratios, you'll notice that in the previous one, what we had was a genotypic ratio of 1 to 2-to-1. So basically you had 1 homozygous dominant, you had 2 heterozygotes, and you had 1 homozygous recessive for a 1 to 2-to-1 ratio. The phenotypic ratio, however, was 3-to-1, 3 freckled to 1 non-freckled. So ultimately, if you were to take a look at a prediction, you would say 3 individuals will have freckles and 1 will not have freckles. However, the problem is each fertilization, that is to say each child, is not influenced by the previous. So for instance, if you have a child in the first round who is homozygous dominant, doesn't necessarily mean that you're going to have a heterozygote, and a homozygote recessive. Right? What that basically means is that you have a force chance every single time you have a kid, you have a 75% chance of having a freckled child. It doesn't mean anything. So you could have 10 kids and they could all have freckles. So each one is only influencing that particular child, that particular birth. So ultimately then, if you take a look at a dominant person, so somebody who's displaying the dominant phenotype, for instance, in this case, the freckled individual, the real question is, how do you know what their genotype is? Because after all we know is they have freckles, which pretty much means that they can either be big F or they can have either big F, right? So that basically will have freckles, or they could also be little f, that could also have freckles. And so in the case of when you have an individual with freckles who reproduces with a woman who does not, the woman we know, right? Because the woman who does not have freckles has to be little f, little f. But the man who has freckles could be either homozygous dominant, in which case all of his kids will have freckles, or it could be heterozygous, in which case half of his offspring or half of his kids would have freckles. So ultimately, if you have a kid who has no freckles, that means they're little f, then that means that the dad has got to be heterozygous. Why? Because mom can only give a little f. That's all she can give. So you know one of those little f's came from mom. Where did the other one come from? It had to have come from dad. So that means dad has to have a little f in there, which means dad has to be heterozygous. That's kind of what it looks like. So in this particular case, if you have, for instance, a homozygous dominant dad with a homozygous recessive mom, then that means all of your kids are going to have freckles because they're all going to be heterozygous. However, if you have a heterozygous dad with a homozygous mom, then that basically means that half of your kids are going to get that recessive allele from dad, which makes them no freckles, and half of your kids are going to get the dominant allele from dad, which means they have freckles. So it's a 1-to-1 type of a ratio. So ultimately, when you take a look at the Punnett square, it can also help you to determine a little bit about these probabilities as you study genetics. And so typically when you're talking about sort of random statistics, you also have to talk about some of the statistical rules. For instance, we have some rules of probability that are at play here in genetics. One is what's called the product rule, the rule of multiplication. And so what this basically has is two different events that are independent of each other. And so you have an outcome that occurs in them simultaneously. And so if you're asking for basically two events to occur simultaneously, then you have to multiply their probabilities together. Okay. And so that's kind of one of the ideas there. So just to give you an idea, let's imagine that I'm flipping a coin in each hand. And I'm going to call one the left hand and the right hand. And so what I want is I want to flip a coin in the left hand and I want it to be heads. And then I want to flip a coin in the right hand and then I also want it to be heads. So the question is, what's the probability of flipping a heads in the left and in the right? For those of you who like word problems, and is the word trigger for multiplication. So for instance, the probability of flipping a heads in the left hand is 1/2, 50-50. In the right hand, it's also 1/2, 50-50. So what's the probability of flipping a heads in both the right and the left? It's 1/2 times 1/2, okay, which becomes 1/4. So that's the product rule. So you use the word and as your trigger. So for instance, the probability of an event in A and in C, that multiplies your probability. We also have another rule called the sum rule, the rule of addition. And this is basically the or term, right? So for instance, what happens if I have a coin in my left hand and a coin in my right hand? And so here's what I want. I want a heads. It doesn't matter which hand I want it in. It can be either in the left or in the right. So my probability states like this. What's the probability of flipping a heads either in the left or in the right? Now, in this particular case, because we have the same probabilities, the probability of flipping a heads in the left is 1/2. The probability of flipping a heads in the right is also 1/2. So that basically gives us 1/2 plus 1/2. And so ultimately, that's the sum rule. So you want an event either in A or in B, you don't really care. Now let's go ahead and take a look at the two trait crosses, right? So basically, both the chromosomes from the mom and the dad separating independently during meiosis one. And so you have lots of different combinations of alleles because of the way you orient yourself in meiosis one. So for instance, if a person had a genotype that is heterozygous for both the F and the S gene, then they would produce 4 different gametes. Now, here's what I want to do. I want to kind of write this out for you so you can see where these gametes come from. And I'm going to put a little slash through the little s so you know which one it is, right? So first of all, number one, each gamete has got to have a representative from the F gene and the S gene, okay? So one choice is to advance forward the dominant for F. Now, when you do that, you can pair it with one of 2 representatives from the S gene. So you can either be big F, big S, that's one gamete, or you can be big F, little s, that's another gamete. Now, what happens if you decide to advance the little f instead? Well, the little f can also be paired up with big S, or that's another gamete, and the 4th gamete could be the little f with the little s. So those would be your 4 gametes. And so they're kind of equal in number. And so when you go through meiosis one, remember, you're basically separating your homologs, and you can basically orient these on either side. So notice how you have, blue versus red versus blue versus blue. So you can orient these on either side, and you're going to sort them in different ways. And so notice how you're segregating them out. You're segregating them out with all this random assortment. And different combinations of these 2 alleles. So when you cross 2 traits, that's what's called a dihybrid cross. Remember, the one trait cross was the monohybrid cross. crossing two traits is the dihybrid cross. So for instance, if it's freckles and short fingers, that's what the S1 is. If a person is homozygous for freckles and short fingers, they're going to be homozygous dominant for each, and then cross them, or you reproduce with a double recessive, so recessive for each who does not have freckles and has long fingers, then the gametes basically are going to be the only thing they give, right? So for instance, the first parent can only give the dominant F and the dominant S. That's going to be one gamete. That's all they can give. And the other parent can only give the little f and the little s. That's all they can give. So their offspring will basically have a combination of both. They'll have one big F from one parent, one little f from the other parent. They're heterozygous for the F gene. The same thing is also true for the S gene. They'll have one big S and one little s. Now then, when these two individuals, say for instance, the heterozygous, which basically will be freckled and short-fingered, if this person were to marry and have kids with another heterozygote, then what you would end up having is the four options, right? So you'd have the two dominant alleles, the dominant recessive, the recessive dominant, and the double recessive. Now, if you combine these guys together, then you would get big F, big F, big S, big S, big F, big F, big S, little s, big F, little s, the big S, and so forth. And so you can see how you'd basically have this kind of 9-3-3-1 ratio, which is typically what we see. So for instance, we have 9 where that is double dominant for both traits, right? So that is basically freckled short. 3 that are recessive, dominant recessive. So dominant for the first trait, recessive for the second. 3 more that are recessive dominant. So now it's no freckles, short fingers, and then 1 that is double recessive. So that is to say recessive for freckles, that is no freckles and long fingers. And so this basically is what we refer to as the phenotypic ratio of a dihybrid cross. It's always the expectation for a dihybrid cross. Now, how do we use probability with the two trait crosses? So we can still use the rules of probability with the dihybrid. And so ultimately, we can approach this as sort of having two monohybrid crosses together. For instance, for freckles, you have a 3-to-1 ratio. And for finger length, you also have a 3-to-1 ratio. So for instance, if we use the product rule, we can calculate the outcome of each one. So for instance, what's the likelihood of having freckles and short fingers? Well, if you have freckles, we already know that it's a 3/4 probability. And short fingers, we know it's a 3/4 probability, which brings you to 9/16. If you have freckles and long, it's basically going to be the probability of freckles times the probability of long fingers, which gives you a 3/16. Same thing with no freckles and short fingers. It's a 1/4 probability times a 3/4 probability, which gives you 3/16. And then no freckles and no long fingers. This is 1 out of 4 outcomes. So it's a 1/4 times 1/4, which is 1/16. Notice you have 9-3-3-1 out of total of 16 options. So you can actually approach this as two monohybrid crosses and multiply them together, kind of like we did with the coin flip experiment. We said we're going to take two independent coin flips and we're going to multiply their probabilities together to see what it would look like if basically they came out at the same time. I mean, like if they came out the way we wanted them to, like with heads and heads. Okay. So a person who has a dominant phenotype, we talked a little bit about this before, can be either homozygous dominant or heterozygous. So for instance, if we have somebody who's homozygous dominant for both the F gene and the S gene, and they reproduce with somebody who's homozygous recessive for both the F gene and the S gene, they'll have heterozygote children who all basically will have the dominant phenotypes. Now, if this heterozygous child crosses with a homozygous recessive, then there is a 25% chance of showing one or both recessive traits. Remember, the two don't have an effect on each other, they're independent from each other. And so ultimately, when you take a look at this, the resulting expectation is to have kind of 1 to 1 to 1 to 1. So essentially, you have a 25% chance for all of them. Why? Because ultimately, just like we saw before in the previous cross, because you have a recessive with a heterozygote, that's going to give you like a 1 to 1 with for each of them. So that's kind of where that comes out. So kind of 1 to 1 to 1 to 1. So it's going to be 1 to 1 for freckles, 1 to 1 for long and short, and the combinations of them. So that's kind of what it looks like. So here is your cross between your heterozygous freckles and short fingers versus your homozygous. Notice your homozygous can only give that one type of gamete. So then ultimately, the only options you have are the 4. So you're either going to be double dominant, you're going to be dominant recessive, recessive dominant, or double recessive. And so it's going to be a 1 to 1 to 1 phenotypic ratio. And so this is just kind of like a summary slide that sort of pulls all that together. Okay. Let's go ahead and take a look at some genetic disorders now. So take a look at genetic disorders. There's a lot of different diseases that can result in mutations. For instance, a mutation is essentially an abnormal gene. It can be passed down from sperm through sperm and egg, just like any other gene. Because this is in the sex cells, it doesn't necessarily mean the parents have any diseases. They could be carrying these mutations around in their genome without even knowing it. And so a lot of these mutations can occur in DNA, and you can come across as different types of things. For instance, you can have what's called an autosomal dominant, where basically all you have to have is just one dominant allele and you're going to have the genetic disorder. Or an autosomal recessive, where you have to have 2 of those recessive alleles in order to see the disorder. And so a lot of times the way we track these through families by using a pedigree, it's kind of like a genealogy chart, only you're tracking genetic disorders. And so this is what allows us to be able to determine whether or not this is dominant or recessive, because if we set up a pedigree of a family and we track the pattern of how it's segregating through a family, that pattern will conform to a classic pattern that these autosomal dominants or autosomal recessive will take on. So, for instance, if you take a look at a pedigree, so typically males are squares and females are circles. A shaded area is somebody who has whatever this genetic disorder is. And so if you see them 2 connected, this is a pairing, this is a mating. So for instance, this individual here had a child with this individual, number 2, to create individual number 3. And so the vertical line is the offspring. So for instance, in this particular case, you have 2 unaffected parents who had an affected child. In the second pattern, you had 2 affected parents who had an unaffected child. And so a lot of times, this is how we can determine whether or not an individual is a carrier of a particular genetic disorder, because typically what will happen is they will carry the recessive trait, but the phenotype is dominant. So for instance, in the autosomal recessive, if both parents are affected, then all the children will be affected, because all they have to pass on is essentially the recessive alleles. And so the children will also be homozygous recessive. This is kind of what it looks like for autosomal recessive inheritance. So here you can basically see you've got an affected male who's a homozygous recessive, an affected female, 2 sibs, sister and brother. And then they also have a sister who's unaffected. Now, all we know about that is that that individual has a dominant allele. We don't know what the other one is. However, because both of these individuals are recessive, that means one of these recessive alleles had to come from mom, and the other one had to come from dad. So that means that at the very least, mom and dad are heterozygous. In this case, that's what they are, because they're not affected. Now, generally speaking, this will tell you that these individuals are carriers. And so you can kind of do this type of analysis and kind of go back and sort of track this back through the family. Now, the important thing also to notice is that notice what you have here is the presence of a double line between mom and dad in generation 3. What that basically means is this is what's called a consanguineous pairing. Inbreeding, basically. And so that basically means that these two individuals were closely related. You can see that from this pedigree, they were first cousins, actually. And so that's a consanguineous relationship. And so what that does is it basically increases the likelihood of these genetic disorders to propagate through the family. And so in some cases, if the condition is dominant, for instance, the parents can be heterozygous and still be affected, right? So for instance, if the child inherits the recessive allele from each parent, they'll be unaffected. But if they inherit at least one dominant allele from a parent, then they'll be an affected individual. This is what it looks like. Notice here, only the unaffected individuals are homozygous. The affected individuals are either heterozygous or they're homozygous dominant. So you can see then that because you have one homozygous individual, that means this recessive allele had to come from one from mom and one from dad. And so that means that even though dad's affected, he's going to be a heterozygote, mom's unaffected, and so she's a homozygous recessive. And so you kind of work back and you can determine what kind of genotype individuals have by kind of working your way back through the pedigree in this kind of analysis. So generally, if you have an autosomal recessive, you have homozygous recessive. So an example of that would be things like Tay-Sachs disease. So this occurs in high frequency among individuals who are part of the Ashkenazi Jewish group of individuals, which is a group of Jewish individuals. I believe they're from a particular region in Germany that is a small bit intermarried, and they kind of accumulated some genetic disorders that are seen higher frequency in that population than in other populations of the human population. And so what causes Tay-Sachs basically is the lack of a lysosome enzyme called hex A, which basically takes out fatty acid proteins in the brain. And so this will kind of accumulate then in the brain, and it can cause blindness and seizure paralysis. We don't really have a cure. Generally speaking, affected individuals will die at an early age, like around 5 or so. So this is kind of what it looks like. So normally in a healthy neuron, you have these lysosomes that are inside the neuron, kind of clearing things out a little bit. And then in Tay-Sachs disease, you'll have like these bulging lysosomes, which are abnormal because they're trying to do what they're supposed to do. They can't do it because they're compromised. Cystic fibrosis is another one. This is really high in frequency in Caucasian groups. This is basically a mutation in a chloride channel. Basically allows you to be able to move the chloride ion across the cell. And so this channel has a really big name, right? The fibrosis conductive transmembrane regulator allele in chromosome 7. So we have a lot of individuals who carry this allele. And so the chloride ion can't pass through this chloride channel, and it starts to accumulate and cause lots of problems. And so this will mess up your water balance. This will mess up your sodium balance. So there's a lot of problems that will come from this. So a couple of things that will happen is it'll start to create this abnormally thick mucus in your bronchial tubes and your pancreatic duct, because it has liver presentations as well, and skin presentations. So there's a lot of different things that will come from cystic fibrosis. And so this is kind of what it looks like. So it's an abnormally formed chloride channel because of the mutation, and so it blocks the passage of the chloride ion, which is something we need in order to balance all of our ions and things like that. Another autosomal recessive is sickle cell disease. So this is one where the red blood cells are sickle shaped, so they're kind of like kind of a hook shape like that, instead of nice and round with that little pucker in it. Generally speaking, the red blood cells are only around for 2 weeks. They should live for about 4 months. 120-ish or so days is about the shelf life. normal red blood cell, but this basically puts a lot of burden on your ability to make these. And this is caused because you have a mutation in a globin gene. So your hemoglobin makes your globin genes basically make your hemoglobin, which is what binds to oxygen in your blood cells. And so you have abnormal hemoglobin, which causes a sickling effect. And so the sickle cells, they can't pass through these narrow capillaries, so you can't deliver oxygen there. It tends to clog up vessels and things like that. And this is the reason why sickle cell is a type of anemia, because you simply can't deliver oxygen and nutrients normally the way you should. It's more prevalent in African Americans. And so this can create a lot of problems. You can treat it with marrow transplants, get some healthy cells in there, blood transfusions, things like that. And so ultimately, in a heterozygote, your normal red blood cells are there. But if you start to get dehydrated, they'll start to sickle. Or if you start to get to oxygen deprivation, they'll sickle. So you can trigger sickling in a heterozygote. You know, that's kind of unusual. It's not like a true dominant in that case. An autosomal dominant disorder that we see is a common one that we see, or not a common one, but a well sort of understood one, is what's called Marfan syndrome. Right, this is basically a deficiency associated with a protein called fibrillin. And so because this is part of your elastic connective tissue, so it'll start to mess with your connective tissue. It's a connective tissue defect, and it's across the board. So it's not just connective tissue and area, it's connective tissue across a lot of areas. So you can get lenses that are dislocated, you get long limbs and fingers, caved chest, weakened wall of the aorta. That's a really bad one that can create cardiovascular episodes. So you can use beta blockers to help with your cardiovascular systems, your corrective lenses, eye surgery. So you get a lot of different sort of connective tissue issues as well. And so a couple of things that you can do is you can get corrective lenses, obviously. Musculoskeletal symptoms could apply. You get osteogenesis imperfecta. Basically, this is weakened or brittle bones, because ultimately this will happen if you have a mutation in collagen, which is a huge component of connective tissue, and also bones. And so this gives us our strength and our rigidity. And so if you don't have that, obviously it's going to be a problem. Another really big dominant, autosomal dominant, is called Huntington's disease. This is a neurodegenerative disease. It's a wicked, wicked disease. Not that any of these are pleasant. But this one's really, really bad. And it's basically because you have a mutation in a gene called Huntington. And Huntington basically, when it's mutated, will clump up inside neurons, render them inactive. And then when they're inactive, often, basically, the neurons will die. And when the neurons die, they don't come back. They don't redivide. So once you lose a neuron, the neuron is gone. And that's what causes the neurodegeneration. You start to lose neurons in important parts of the brain, and that basically starts to create massive neurodegeneration. It's a late onset genetic disease. However, an individual could live a relatively normal life well into their 30s or even into their 40s, and then they'll start to get some muscular sort of discoordination. Their gait, which is oftentimes associated with muscular discoordination, will start to become a little off. They'll start to get dementia as different regions of their brain begin to erode due to neuronal death. And so after the initial onset, you've got maybe 15 to 20 years. And so one of the reasons why this is a particularly wicked disease, Huntington, so here you can see neurons in a normal brain and then loss of neurons in a Huntington's brain. So you can see just how much actual neural loss is associated. So this is massive neural loss. And so the reason why Huntington's is a particularly evil is because of its late onset. Because a lot of times, by the time you start manifesting symptoms, let's say in your 40s, oftentimes you've already had kids. And since it's a dominant, that means half of your kids are going to have the same disease that you have. So it's not uncommon for this particular disease to actually cut across and devastate not just one generation, but multiple generations of family. Chances are that that 40-year-old with Huntington's got it from one of their parents who has Huntington's. Chances are that they had to go through a very, very difficult stage with their own parents, where they had to take care of them because they saw their parents basically in the prime of their life, their 30s and their 40s, basically start to neurologically fall apart. And they had to do some very difficult work in caring for that parent and literally just watching their parent just fall apart in front of them, and then ultimately die early 50s, maybe 60s. And then here they are now at about the same age as their parents were when they first got the symptoms manifesting. And then they realize that now they also have Huntington's, and they're going to have to go through the exact same thing that they saw their parents go through. And not only that, but now they're going to basically inflict the same difficulty and hardship and heartache and trauma on their own kids. So it's kind of like you see your future in the ugliness of what your own family is going through. And not only that, but you see that and you basically say, this is what I'm going to get. And then you turn around and you say, this is the exact same thing that half of my children are going to get. So it's unlike most diseases, this one really has a wicked way of just cutting across multiple generations of family, just devastating them. It's a really, really just debilitating neurological disease. So let's take a look at a couple of other types of ways of inheriting genes. So we talked a little bit about one trait inheritance, two trait inheritance. Now let's talk about what's called polygenic inheritance. Polygenic inheritance is a situation where a trait is controlled by multiple sets of alleles. So there's a lot of genes involved in it, not just one. Good example of that is height and skin color. Now, because you have lots of alleles involved, both dominant and recessive, they tend to be additive. And when they're additive, they create what's referred to as continuous variation. So basically, you're going to see very few that are extreme on one side, recessive, and you're going to see very few that are extremely dominant. And most individuals are going to have combinations in the middle. Looks like a bell curve, right? So that bell-shaped curve is what creates this continuous sort of distribution. Now, there may be environmental effects that also adds to that as well. But for the most part, the traits are continuous. And this is kind of what it looks like. If you take individuals and you have them line up by height, they're going to have a few people that are very, very short, a few people that are very, very tall, but majority of people are going to be somewhere in between. It's a bell curve, and it's a continuous trait. So that's polygenic. The same thing can also be true for skin color. So skin color basically is produced by cells called melanocytes. That's what they do. They basically produce your pigment and they deposit it into your skin. There are some 100 different genes that affect skin color. And of course, it's influenced by sunlight, so there's an environmental piece as well. But ultimately, the phenotypes can range, right? So for instance, the more of the light alleles that you have, then you're going to be on this end of the spectrum. The more of the dark dominant alleles that you have, you're going to be on this end of the spectrum. The majority of people have a mixture of the two. So for instance, you can see the majority of people are going to be somewhere in between with an even mix of light and dark alleles. And some of them may have a few more light alleles. Some of them may have a few more dark alleles, but for the most part, the overwhelming majority are going to be somewhere in the middle. And that's kind of what we see with skin color, right? It's very rare to find very, very pale people. It's very rare to find very, very dark people. Most people across the globe, if you take a look at the entire human population, are going to be somewhere in the middle, kind of olivish leaning dark, olivish leaning light. Okay, and that's just because it's a polygenic trait. Now, this also leads us to what's called multifactorial traits. So oftentimes they are going to be polygenic, but they're also going to have strong environmental impact as well. That's why they're called multifactorial. There's just a lot going on there. Right? A good example of this would be like, for instance, cleft lip, or the way that your palate is forming. Schizophrenia, a lot of mental health disorders. Schizophrenia, diabetes, allergies, cancers are definitely multifactorial. That's the reason why it's almost impossible to come up with a cancer cure. But if you take a look at the Himalayan rabbit, this is a good example of the environmental influence on an animal. So for instance, you have a dark pigment deposition on the ears, the nose, the paws, and the tails. And so that's because there's an enzyme that makes melanin that's temperature sensitive. So for instance, in cold temperatures, it's going to deposit melanin. That's the reason why you get these melanin deposits in these cold extremities. And then in the warmer areas, like the core body temperature area, it doesn't deposit the melanin because it doesn't make it. So that's an example of how environment can basically affect the phenotype. Now, pleiotropy is kind of a difference. Basically, where you have a single gene or single mutation, a single gene, but it's basically going to create lots of traits. So multiple traits. So two or more distinct traits. So Marfan syndrome is an example, right? So essentially, if you take a look at Marfan syndrome, you have a connective tissue defect. That's just one gene. But you're going to have multiple manifestations. For instance, you're going to have a set of symptoms in the skeleton. This is one phenotype. You're going to have cardiovascular symptoms. That's another phenotype. So that's phenotype 2. You're going to have a situation with the eyes, right? With the lenses of the eyes. That's phenotype number 3. The lungs are going to have a phenotype. So that's a phenotype number 4. And then, of course, you have a skin phenotype. That's phenotype number 5. So you have 5 different phenotypes from one gene. That's pleiotropy. Now, there's also a couple of other ways of inheriting genes. There's what we call incomplete dominance. So this is another one. So this is a situation where the dominant does not completely mask the recessive. So hypercholesterolemia, familial hypercholesterolemia, is associated with a gene that controls the number of LDL cholesterol. Those are your bad cholesterol, the sticky one that gets stuck in your arteries. And it controls the number of LDL cholesterol receptor proteins in the cells that have your plasma membrane. So basically, a person with these 2 mutated alleles lacks the LDL receptors. And so as a result, it tends to stick. In the arteries. And so oftentimes these individuals will have cardiovascular disease and they can die before the age of 30, very young. And so they're developing these cholesterol deposits in the skin and the tendons and the cornea. It's basically everywhere. So you don't really have an LDL cholesterol protein to manage your lipids. A person who has only one has half the normal receptors. They may still die young, but basically they may get to a little deeper into life. So they're still kind of limping around. They're still kind of compromised, but not quite as bad as a person with 2. Now, with 2 normal alleles, they don't have the familial hypercholesterolemia, okay, because they have everything they need in order to manage these sticky lipids. And so this is kind of like what this looks like. So essentially, if you have 2 normals, you're going to have a normal situation. If you're heterozygote, it's going to be a little bit more problematic. It's going to be a little stickier. You're still going to have cholesterolemia, although not quite as strong. If you're homozygous, then you're going to have literally no lipid management, and you're going to see these huge cholesterol deposits throughout your body. So codominance is another version of kind of an inheritance pattern. This is basically where the alleles are equally expressed with each other. So the most common example that we use actually in genetics is the ABO blood group. So the red blood cells essentially have both an A type and a B type blood. And so as a result, what happens is these 2 are dominant to each other, right? So that's basically what we have. So these are both dominant to each other, dominant. And they share equally in the phenotype. We'll take a look at this here in just a second. But the other one, which is called multi-allelism, is another type of a pattern where in the human population, a gene has more than 2 alleles. For instance, the ABO blood group is a good example. In the ABO blood grouping, you have 3 different alleles. So you've got the dominant A. So the big I stands for immunoglobulin, and the superscript A is the A type. And then there's B, and then there's little I, which is recessive for these two. And so you have different types of genotypes then for a person with A type blood and also for B type blood. So if you kind of put them all together, then you've got both multi-allelism and codominance together. So for instance, in a person with A type blood, they can genotypically either be homozygous dominant, big A, or big A, little I, so heterozygous. That basically means that A is dominant to little I. A B type person can be either homozygous dominant, big B, or big B, little I. And so that is basically big B is dominant to the little I. Now, an AB person has both the dominant alleles, this is codominance, and they're equally sharing together in the phenotype. The O type person has little I. Now, if you're wondering, basically what happens is with these ABO blood groupings, all they are is essentially your red blood cells have on their surface these glycoproteins. As a matter of fact, there's lots of glycoproteins on all of your cells. The only problem is this is the only group of glycoproteins, that the RH, that basically creates an immune response. So for instance, if an A-type person will have an A-type carbohydrate on their glycoprotein, a B-type person will have the B-type carbohydrate on their glycoprotein. An AB person will have one of each, equally shared. An O-type person will have neither. So it's just basically a matter of what kinds of carbohydrates you have out there. So if you take a look then at some of the genetics of blood typing, so in this particular case, you have a B-type parent who is homozygous, or excuse me, heterozygous, and paired with an A-type person who's heterozygous. Then the A-type mother is going to either produce the big A or the little I. Those are the two gametes, the two eggs that she can make. The dad's going to make the big B or the little I. Those are the two sperm that he can make. So if you get a crossing of these two first, you're going to get an AB baby. If you cross these two, you're going to get a B type. These two, you're going to get A type and then an O type. So you're going to basically get 1 to 1 to 1 to 1. That's basically what happens when you have this particular pairing and this particular mating. So that just kind of gives you an idea of some of the pairing possibilities associated with multi-allelism with codominance going on at the same time. So let's take a look at sex-linked inheritance. All right, so what about inheritance with the X and the Y? We've been talking mostly about the first 22, right, the autosomes. So when you take a look at males and females, we have 23 pairs of chromosomes. The first 22, 1 through 22, are what's referred to as autosomes. That's the reason why we're talking about these as autosomal dominant, autosomal recessive. So that's basically the first 22. The other two are going to be the sex chromosomes. That's the X and the Y. So for instance, males will have an X and a Y, females will have 2 X's. So Y basically has the gene that is the male determiner for the male gender. So the Y confers maleness in humans. Now, if you have a sex-linked trait, ultimately, these are going to be associated with genes that are on the X and or, to a lesser degree, the Y. Most sex-linked genes are on the X because there's a lot more genes on the X than there are on the Y. And so, ultimately, the X will basically affect both males and females because both males and females carry an X, whereas only the males carry the Y. So ultimately, the male will always receive his X from his mom. And so the Y chromosome, however, is always coming from the dad. So ultimately, the dad can either pass forward the X or the Y. But if the male has a Y, then he got that from his dad. There's no other way he could have gotten that, okay, because the mom doesn't have a Y. The Y chromosome is important because it doesn't have the same genes as the X chromosome, so they're different. In terms of the genes that they carry, they're very different. And so usually, most of the time, not always, the sex-linked disorders are recessive in nature. And so, for instance, for a female to be affected by a sex-linked inheritance, she has to receive 2 alleles one from each parent in order to have this disorder. As it turns out, that usually is a rarity, right? So for instance, if you take a look at color blindness, and so this is an X-linked trait. So here we have big B, which is normal color vision, whereas little b is going to be colorblind. Normally speaking, the homozygous dominant female is normal color vision. The heterozygous female is a carrier female who has normal vision. The homozygous recessive is a female who's colorblind. That's actually fairly rare. And then the males, which are referred to as not as homozygous, but they're referred to as hemizygous because they're half. These guys will basically be 50-50. So you'll have half of them are going to be hemizygous dominant with normal color vision and hemizygous recessive with color blindness. And so ultimately, most females for sex-linked genes are carriers, but they usually have the normal gene. So the mother herself is normal, but she can pass on the allele, the mutated allele, to her sons and also to her daughters as well. And so for a female to be colorblind would require her to receive that allele from both parents, which means that essentially her dad would have to be colorblind and her mom would have to be a carrier. And that's the reason why colorblindness and X-linked traits are typically more common in males, is because they only need one recessive in order to be able to see it because of the Y chromosome not being the same, having the same genes as the X chromosome. So your color blindness and all X-linked traits basically are going to be coming from the mother because the son always gets the X chromosome from the mother, gets the Y from dad. So that's kind of what it looks like. So if you take a look then at color blindness, so here is basically a normal color dad with a carrier mom. So the dad can either give an XB or an X little b. Mom can either give an X big B or Y, rather vice versa. And so what happens then is if you take this egg with this sperm, you're going to get a homozygous dominant, that is normal color vision, and it's going to be female. And if you combine this egg with this sperm, you're going to get a heterozygous carrier female. If you combine this egg with this sperm, you're going to get a normal colored male. And then this egg with this sperm, you're going to have a colorblind male. So in this particular situation, all of the females are going to have normal vision, and only one of them is going to be a carrier. Of the males, you have half and half. So one is going to be colorblind and one is going to be normal vision, and that basically is reflective of what mom had. So half of her chromosomes were colorblind and half were normal vision, and that's basically what happens. So all males essentially have like half of what mom has. So so this affects males because of that recessiveness, and so because Ys lack the chromosome, and so oftentimes they'll pass from grandfather to grandson. So like for instance, an X-linked recessive condition will go from grandfather. It'll go because the grandfather gives the Y to his son and the X to his daughter. Then that X will go through a daughter who's likely going to be a carrier, who will then have to give it to her son. So this is the reason why it skips generations, is because the daughter of the male is a carrier and she passed it on. So here's a good example of what's happening out here. So here's our grandfather, who happens to be colorblind, who grandma is normal, homozygous dominant. So when he passes on his Y, he's passing it on to his sons. And so that Y is going to his sons. So ultimately the sons are reflecting what grandma looked like. However, when he passes on his X, he's passing it on to his daughters. So his daughter, he'll give a colorblind X to his daughter, and then that daughter gets a normal one, and she's a carrier. Now, when she basically has, whoops, sorry, he didn't pass that one X on that because that would be inbreeding. So when she goes and has a kid, then she passes that colorblind X that she got from her dad on to her kids. And that's basically where you get the grandson coming out with the colorblind X. But she's also going to produce a carrier daughter as well. And she'll produce a normal vision son and a homozygous dominant. So the mother will basically produce both. And so you typically see this sort of skipping generations in X-linked recessives. Now, what about an X-linked dominant? So ultimately, when you have a male with this condition, this basically means the male is passing this on to his daughters. And so you have a 100% chance of having the condition. And so the female can then pass this X-linked dominant to both of her sons and her daughters. So this becomes basically just like, I'm just going to hit you in every single generation. And so if you're colorblindness, which some of you may be familiar with or not, basically is simply just a deficiency in some of your cones, your ability to discern between different colors. For instance, oftentimes there's what's called a red-green deficiency. So you can't really see reds and greens. They just kind of look like shades of gray. There's no treatments because it's a genetic defect. There's typically no treatment and there's no cure for genetic disorders. So this is actually fairly common. So it's about 1 in 12 males, about 1 in 200 females. So it's actually fairly common. Now, when you take a look at another X-linked recessive, you have Duchenne's muscular dystrophy, which is a degeneration of muscles. So you kind of get this sort of gait. You fall a lot because of muscular discoordination. You have difficulty sort of maneuvering your muscles. Oftentimes, you'll end up in a wheelchair at an early age. And then usually you get so much muscular dysfunction that you can't actually maintain core visceral systems and you die early in your 20s. Generally speaking, affected males rarely have kids. And so what happens is the carrier mother basically passes it from her to her daughter. That's usually how the allele stays in the population. So this is kind of what normal skeletal muscle tissue looks like. And then when you get muscular dystrophy, you can see how you have a loss of muscle tissue. And so you have now the cause of this is a mutation in a protein called dystrophin. And so ultimately what happens is calcium leaks into the cell, which basically stimulates an enzyme to basically dissolve muscle fibers. So you're kind of self-digesting muscle fibers, and that's kind of where the muscle atrophy comes into play, kind of a wasting away of musculature. Another type of X-linked recessive that we have is Fragile X. This is an interesting one. This is will basically create an inherited kind of an intellectual disability, so cognitive developmental issues. And so this will kind of create kind of what's called the autism spectral disorder, ASD. And so some of the things that you'll see is obviously different types of mental and cognitive abnormalities, prominent jaw, flexible joints, genital abnormalities in the production of the genitals, typically a strong aversion to being touched or making eye contact, speech patterns, things like that. So just general types of things that are associated with kind of cognitive disruption and communication. Hemophilia is another recessive, X-linked recessive. This was a common one that we found out early on in the early part of the 20th century. And so there's two different types, hemophilia A and B. So in A, it's the absence of clotting factor 8, and in B, it's the absence of clotting factor 9. And so ultimately, you have blood clots, so it's basically too thin. And this causes bleed outs. So you can hemorrhage. And so over silly things like just a minor cut or even a bloody nose can be a life-threatening situation for a hemophiliac. And so you can stop the hemorrhages by getting transfusions of fresh blood, getting some of that clotting factor into you so that your blood can coagulate and clot normally. But this is a genetic disorder, an X-linked genetic disorder. Okay, so that is the genetics chapter.